So what we're going to do next is we're going to learn the different stages that the cell goes through when it's undergoing division to make these haploid gametes. So remember, we're trying to go from a cell that has 46 chromosomes, and not only 46 chromosomes, but it's 2 of every chromosome. 2 number 1s, 2 number 2s, 2 number 3s. We're trying to go from that down to a cell that only has 23 chromosomes, and those 23 are very specific. It's only 1 number 1, 1 number 2, 1 number 3, 1 number 4, etc. So we looked at this slide in lecture and we said we're going to pick 23, one of these, one of these, one of these, one of those, all the way up to the top, right? So to get that done, we'll do a sperm first. We're going to say we're going to model this in a testes, so we're going to make a sperm cell. So because we have a lot of sorting and organization to do with these chromosomes, we're going to use some of the machinery that we already have set up in our cell for doing regular mitosis with a T. Right? We're not doing mitosis, we're doing meiosis, but we're going to use some of the parts that we use for mitosis because we already have them. And the first thing that a cell does when it goes through regular mitosis is that it's going to replicate all of its DNA, and it's going to shrink down the DNA and condense it into short fat chromosomes, right? So we're going to go through these stages. I'm going to show you a cell here. This cell on the document camera. All right, here's my cell. These are the chromosomes inside of my cell. Okay, so my first, and this is a testes cell. My first question for you is this cell a haploid cell or a diploid cell?
Diploid.
We have 1 vote for haploid and 1 vote for diploid. Who thinks it's diploid? Okay, who thinks it's haploid? Oh, all right, okay. How are you going to tell? What are you going to look for to decide whether it's haploid or diploid?
Haploids have 1 from each pair.
Okay, haploids have 1 of each type of chromosome. Diploids have 2 of each type of chromosome. So looking at the cell, do we have 1 of every kind of chromosome or 2? 2, right? There's 2 long yellow ones and 2 short red ones, right? So this is definitely, currently a diploid cell, right? So we would say it is 2N, right? Instead of having to write out the whole word diploid, we just write out 2N because we're lazy and don't like to write words in science, right? Not really. So we say this cell is 2N. Now, how many total chromosomes do we have on this slide right now? How many chromosomes are sitting there? 4. So we can say its diploid chromosome number is 4. That's how you would write that. 2N equals 4. If this was a human cell, I couldn't fit it on my thing, it would be too busy. But if it was a human cell, what would we have? 2N equals, for humans, 2N equals 46, right? This is for humans. Obviously, I can't really, if I put 46 pieces of yarn here, it would be a big old mess. So I'm just going down to 2 to make it simpler. Okay, so the stages of meiosis, just to confuse you, have the same names as the stages of mitosis, which isn't very helpful, but it is what it is. So the stages are called interphase, and then we have prophase, metaphase, anaphase, and telophase, just like we had in mitosis, followed by cytokinesis, so that cell divides. But because in meiosis we're doing 2 divisions, we're going to divide the cell twice, we have to do this whole thing again. So we're going to go through another prophase, metaphase, anaphase, and telophase, followed by a second cytokinesis. So we go prophase, metaphase, anaphase, telophase, split, and then we do another prophase, metaphase, anaphase, telophase, split. So we go from 1 cell to 2 cells, and then we go from 2 cells to 4 cells. So from one starting cell in the testicle, we can get 4 sperm out. 1 leads to 4 in this situation from making sperm. So that we know the difference between the different, whether we're doing the first division or the second division, we put numbers here. So we call this prophase 1, metaphase 1, anaphase 1, telophase 1, followed by prophase 2, metaphase 2, anaphase 2, and telophase 2. And you've got to put those numbers. Because if you just write prophase without prophase 1 or prophase 2, I don't know A, which one you're talking about. And if you write just prophase, I'm going to think you mean mitosis with a T. Because it's just prophase, no number, in mitosis. You see the difference? So when you're talking about meiosis, you've got to have the numbers after them. 1 or 2.
As well as for cytokinesis.
No. Cytokinesis isn't really a stage, it's just something that happens. All right, so let's get to our little diagram here. Go back to this. So here's my cell. It's diploid. It's got 4 total chromosomes, 1 pair here and 1 pair here. Okay, so the first thing that happens in meiosis is that we are going to replicate the DNA and then condense it down. So when we replicate the DNA, each chromosome is going to end up looking like an X. So now I have 4 replicated chromosomes. I replicated each individual one, so now I've got 4 X's. Remember that? Okay. So right now we're still in interphase. We're getting everything ready. We're using the machinery we've already got that we know how to use because we do it for regular DNA replication. So we're going to copy all the DNA, make all the chromosomes look like these replicated X's. And then what's the next thing we're going to do? To make these things easy to handle, what are we going to do with the DNA? Condense it, right. Because right now it's long and floppy and it's going to get tangled up. So to make it easy to handle, I'm going to, ta-da, turn it into replicated chromosomes. All right. So right now we're at the end of interphase. All the chromosomes have been replicated and they've all been condensed. All right, next we're going to move into prophase 1. This is where it starts getting a little different. Actually, a lot different. Okay, so in prophase 1, we're going to do some of the things we're already familiar with. We're going to destroy the nuclear envelope and we're going to build a spindle. Okay, so destroying the nuclear envelope, building a spindle. Remember the spindle, the fibers that stretch across the cell? That's so far so good. But now something different is going to happen. Different to mitosis. What's going to happen is the 2 chromosomes that are a pair, this one and this one, they're going to find each other and they're going to snuggle up with each other like this. And this pair is going to find each other and they're going to snuggle up with each other like this. So my pairs of replicated chromosomes are finding their partner and they're lying side by side. This did not happen in mitosis, right? This is new. This is a new thing. So we say homologous pairs come together. Do you remember we talked about homologous pairs? The two pairs of the number 1 and the number 1 come together. Right. So when they do this, this is called synapsis. Synapsis. And I'm going to flip over to the PC here. So here they're showing the two chromosomes that are the same, that this is one of the chromosome ones, this is the other chromosome one. It's replicated because they look an X, but they're coming together. This is called synapsis. When they come together like this, they then entwine the arms of the chromosomes. And this is called crossing over. So on mine, can you see how they're so tightly attached to each other that the arms, otherwise known as the chromatids, hold on, takes a while, there you go. The chromatids are twisting up with each other. And the arms of this one are twisting up with each other. That's called crossing over. I'm literally taking the arms of the X's and they're tangling with each other like this. Crossing over. This is all happening in prophase 1. There's a lot of action in prophase 1. Okay. When they do this, they actually tangle up so tightly. The arms of the homologous pairs tangle so tightly that they physically break themselves apart. So this arm and this arm get so tightly wrapped around each other that the backbone of the DNA breaks. It's like you're breaking your chromosomes. This is like big deal. And when they break, then they try and rejoin. They don't like being broken. They're trying to rejoin, but sometimes they rejoin to the wrong arm. Like this red one used to go to this red one, and this blue one used to go to this blue one, but now they've tangled themselves up so much and they've snapped. When they grabbed hold back to the backbone again, they grabbed hold of the wrong piece. And so now what's going on here is you've got this arm is now attached to a piece of the arm from this blue one. They've kind of gotten mixed together, patchwork together. This whole process is known as synapsis followed by crossing over. It's super important. So you end up with a chromosome that's kind of patched together from pieces of two chromosomes. All right, now, when you do that, when you break chromosomes like that, it's a bit of a high-risk strategy, right? When you're breaking chromosome arms, you're hoping that they're going to join back up together and you can recreate the chromosomes that you snapped, but sometimes it doesn't go right. Sometimes when they join back up together, they grab a wrong piece or something different, and this can result in mutations. So these mutations aren't the kind of mutations that we talked about in Unit 4. In Unit 4, we were talking about like a little change in the sequence where it affects a single protein, right? We're just talking about one little pinpoint change. These kind of mutations are giant chunks of whole chromosomes getting misplaced. So these mutations are going to be pretty major when they happen. And there's a range of different things that can go wrong. 4 different types: deletions, duplications, translocations, and inversions. So there's an example here of each type of mutation. See if you can line up which one goes with which. Look at the words and see if you can figure out what's going on with our chromosomes there. See if you can make sense of that. Let's see, let's go through these. Which one do you think is the deletion? The third one? Yes, the third one down, look, because it's supposed to go, this chromosome goes, these are the genes, A, B, C, D, E, F, G on the chromosome, and somehow it lost its top bit, right? Lost A. So we've deleted a chunk of this chromosome. It crossed over, but it didn't manage to grab hold of another piece, so it just lost a piece of itself. That's a deletion. Which is the duplication? At the very bottom, right? This is our duplication. Somehow we've got A, B, C, D, E over again, and then F, G. So we've added a piece in here by accident. We've also got an inversion, which is the inversion. Right, a piece has got put in backwards or upside down. Instead of going A, B, C, D, it goes A, B, D, C. That piece got put in upside down. All these things can happen when you're chopping and trading chunks. And then the last one, the translocation, I left that to the end because obviously this is a translocation by elimination. So what's going on here? If we look here, we have this chromosome and they've traded chunks down here, right? Isn't that what we're supposed to be doing on this? Aren't we supposed to be trading chunks between these arms? Yeah? But what's happened here is I've traded chunks from this chromosome with chunks with a chromosome that isn't a homologous pair. This is a whole different chromosome. This is like chromosome number 1 trading chunks with chromosome number 7. Chromosome 1 is supposed to trade with another chromosome 1 because chromosome 1 pairs up with 1 and 1 trades with 1. So if you trade a D, you should trade a D for another D. But here we've traded a F and G for a P and Q, for a Q and R. So this would be like chromosome 1 accidentally getting tangled up with chromosome 7 and trading a piece. Well, you can't do that. 1 gets tangled up with another 1. 7 gets tangled up with another 7. 7 can trade chunks with 7. 1 can trade chunks with 1. But here we've traded between 2 different chromosomes, and that can't happen. So this is another example of the kind of mutation. So obviously these are serious things. These are going to make majorly messed up chromosomes. Probably messed up to the point where they're going to be unsurvivable errors. If you were to inherit a chromosome like this, you'd probably die or not even be born. So why do we do it? Why do we allow this to happen when we make eggs and when we make sperm? What does it allow us to do? The reason we allow this is that it causes diversity. It creates new versions of existing chromosomes, and it makes everybody in this room be different from every other person in this room and every other person on the whole history of planet Earth. Okay? Because what you're doing, if you're doing this right now in your testes if you're a male, or in your eggs if you're a female, if you're a female, you did it a long time ago, but when you did this, you took the chromosome 1 you got from your mom, and you took the chromosome 1 you got from your dad. You replicated them so they look like X's. You paired them up, and then you traded chunks with them. So you created a brand new, never before seen chromosome 1 that has some chunks from your mom and some chunks from your dad. And you did that in your ovaries. So your ovaries are really taking the genes that you got from your mom and dad and mixing them up into new combinations, and then you can pass that on to your child. But you're not really mixing up your genes, you're mixing up the genes that came from your parents, so the potential baby's grandparents. Does that make sense? All right, so if we relate that back to my slide here, all right, so here's a pair of chromosomes right here. This one came from, say, my mom. This one came from my dad. I replicated them, so now they look like X's. And now I'm trading chunks between the one that I got from my mom and the one that I got from my dad. So this arm of this chromosome is going to be a mixture of genes from my mom and genes from my dad. If this goes into my baby and becomes a baby, then that baby is going to have some of my mom's genes and some of my dad's genes, or the baby's maternal grandmother's genes and the baby's maternal grandfather's genes mixed together in that baby. All right? Making a little bit sense? So your ovaries or your testicles are places where you shuffle up the genes of your parents to pass them on to your offspring. And you're shuffling them up in a way that is never before seen on the whole history of planet Earth. Nobody has ever put those two sets of genes together ever before, and they never will again. It's happening for the first time and the only time ever in you, in your ovaries or your testicles, in that egg or that sperm. And you'll never make the same egg again, you'll never make the same sperm again. And that's why you're not the same as your brother or sister, even though you came from the same parents, thank God, right? So a different egg and a different sperm made you than made your brother or made your sister. And so even though you have the same parents and the same grandparents, the way it was all mixed together was different in each case. So you're not exactly the same as anybody. although you do share traits with these people. All right, what do you think? Make sense? We'll go through it a couple times more, but for now we'll leave it at that. So that's prophase. All right, so prophase 1. So once again, destroy the nuclear envelope, create the spindle, pair the homologs together, and do synapsis and crossing over. There's a lot that's going on. All right, gets easier from here on out. So next up, we're going to do metaphase. What generally happens in metaphase? What does the M stand for? Middle, right? So in metaphase, we're going to take our homologous pairs, and we're going to line them up in the middle like this. Metaphase 1, right there. Pairs of homologs, I could say pairs of replicated homologs, are lined up in the middle of the cell. What do you think is going to happen in anaphase? Action, right? What happens in anaphase? Pulling apart, right? What I'm going to pull apart are the homologous pairs, not the individual X's. The X's are staying together. Look, there's an X here, there's an X here. But I'm pulling apart the pairs. There we go. That was anaphase 1. So my homologous pairs of replicated chromosomes are pulled apart. Telophase, basically we're going to relax the DNA a little bit. And we're going to do cytokinesis. We're going to split into 2 cells. So we're right here. So we've been through all these steps, okay? All right, so now let's look at this individual cell on the left side. This one here. Is this cell haploid now or diploid? Does it have 1 of each kind of chromosome or 2 of each kinds of chromosomes? It's a hard question, right? Because it's replicated. It's not. It's only 1 of each kind of chromosome. This is replicated, but it's not pulled apart yet. So it's 1 chromosome, and this is 1 chromosome. So it has 1 big one and 1 small one. Now it's haploid. And we've gone from 4 chromosomes to only 2. It's replicated because it's an X, but it's still 1 chromosome because it's still held together. Okay? So this is the end of cytokinesis. We're going to start stage 2 now, the 2nd division. All right, so prophase 2 looks a lot like telophase 1. There's not a lot of difference. We're going to make sure everything's all coiled up. And we're going to build a new spindle because we're going to do division in these cells. So we're going to build a spindle going this way. So build a new spindle. Metaphase 2. What do you think I'm going to do in metaphase 2? Once again, line these guys up at the middle. That's metaphase 2 right there. Anaphase 2. What do you think I'm going to do in anaphase 2? Pull them apart, right? Anaphase is always action. Ready? Going to pull apart this. Pull apart this. Pull apart this one. Pull apart that one. So I pulled apart these this way and these this way. I was anaphase 2. Telophase 2, the DNA is going to relax. Back to what it was before. Let's do 1 cell. And then cytokinesis, we're going to divide again. Divide here, divide here, divide here, divide here. And now I've created one cell here, one cell here, one cell here, and one cell here. There's my 4 cells. So we start off with 1 cell in the testes. I've made 4 sperm. 1, 2, 3, 4. And each sperm is haploid. Look, it only has 1 of each chromosome in. 1 red one, one short red one, 1 long yellow one. Four haploid cells from 1 original diploid cell. All right, I'm going to go through the whole thing from the beginning. Let's see how we do. All right, get a fresh cell. So once again, here's my testes cell, hanging out in the testicle. It's a diploid cell. It's got 2 of everything, but it's only got 4 total chromosomes, so it's small. But we've got 2 of everything. Diploid cell to start with. So what's the first thing we're going to do to the DNA in interphase? Replicate it, right. So replicate, 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 replicate. Excellent. Now it's replicated. And we're also going to do what? Condense it. Excellent. Makes it easier to handle. There we go. Ta-da! All right, interphase is done. Prophase 1, what are we going to do? Destroy the nucleus. Build the spindle. Right? Imagine that's all happening in your head. What are we going to do with our chromosomes? So they're going to snuggle up. They're going to be, where have you been all my life, number 1? Oh, there you are, number 1. Oh, there you are. Now, if this is happening in you, this will freak you out, but it's number 1 from your mom meeting number 1 from your dad and getting all cozy. And number 2 from your mom and number 2 from your dad in your testicle meeting each other and getting all cozy. All right? That's the only time that they pair up, is during meiosis. So it's not happening in all the cells of your body, only in your ovaries if you're female and in your testes if you're male. Right? But the number 1 from your mom meets the number 1 from your dad, the number 2 from your mom meets the number 2 from your dad. And they snuggle up. What's this called? These are called homologous pairs, and they're doing synapsis. Perfect, yes, synapsis. And once they've done synapsis, they're going to cross over. What happens in crossing over? They're going to tangle up, and they're going to trade pieces, which hopefully doesn't go wrong, but sometimes does. All right. But it's worth it because of the diversity, because you don't want to be like your brother, and you don't want to be like your sister, right? Right, okay. So we're at the end of prophase 1. Metaphase 1, what am I going to do? Line them up, line the pairs of homologous replicated chromosomes up at the metaphase. Let's do it this way. At metaphase. Metaphase 1. Anaphase 1. Action. Ooh, there they go. There they go. All right. Telophase 1. Just going to kind of relax, do their thing. Cytokinesis. Two cells. Pinch apart. All right, excellent. Prophase 2. Build a spindle, do your thing. Metaphase 2. Line them at the middle. Anaphase 2. Pull apart, this time we're pulling apart the individual replicated chromosomes, separating the chromatids. Ooh, there they go. Anaphase 2. Telophase 2. they're going to relax. You're going to make your new nuclei. And then cytokinesis, separate, separate. All right, so that gives us sperm number 1, sperm number 2, sperm number 3, sperm number 4. And each one is haploid, only one of each chromosome. Good. That is the steps of meiosis. I think it's much easier to do it looking at something moving than just pictures. I hope that made sense.